Hello and welcome. My name is Andrew Berry. I teach evolutionary biology and genetics at Harvard University. And it's my great pleasure and honor to be here. Well, not to quite be here, but it's my great pleasure and honor to be invited and to present to the Evolution Day event put on by the Acıbadem Genetics and Molecular Biology Club. So, yes, apologies, I'm not there in person. I hope this will do instead. So what I'm going to talk about is, well, I'm just going to introduce very briefly the theory of evolution in general, and then I'm going to talk about the impact that ancient DNA has had on our science. So it's going to be simple, it's going to be sweet, it's going to be short, and I hope it's going to be illuminating. But anyway, let's launch this presentation. Good. So my title is Ancient DNA, Evolution's Time Machine. There's my email address. Please don't hesitate to get in touch with me if I can be helpful in the future about evolution, about biology, about, well, anything else. But let's talk about the theory of evolution by natural selection. As I say, I think this, well, I'm biased. I'm an evolutionary biologist. This is the best idea anyone has ever had. End of story. Why? One, it's a super simple idea. So you don't need to have a PhD in statistical mechanics to understand it, for example. It's a very straightforward idea. It's a simple idea. And yet it explains so much. So critically, amongst the things it explained is biological diversity. I don't know how many, it's still open to debate how many species inhabit planet Earth at the moment. I think the best estimate in my book is around 15 million. Extraordinary biodiversity, which I've enshrined with this sort of crazy looking marine organism on the left. It's sort of loony in a way. But it's emblematic of what has been achieved in approximately 3.9 billion years. If life originated 3.9 billion years ago, then we've had this ramifying tree building process as new lines of life have split off in the tree of life. And what you've got is what we have today, which is just stunning and spectacular. But not just that. The second thing is all those living species, all those organisms are beautifully, exquisitely adapted to their environment. So I've got a hummingbird here. Its bill is just the right size and shape to probe to the bottom of that flower to get the nectar that it's after. And that, of course, is we've got change occurring through mutation over generation, over generation, over generation. And certain mutations are being eliminated, certain mutations are being favored. That is natural selection, the gradual generation by generation process that creates adaptation. But I think unequivocally the most significant, important, and, well, seismic part of this theory is this. It explains us. I've got Shakespeare. Maybe you as Turks don't agree that Shakespeare's the greatest human being on all time. But anyway, I'm English. I'm allowed to make that assertion. No, this simple idea explains us. It explains who we are. It explains where we came from. And I do think this is the biggest, most significant idea in the history of ideas, that us, brilliant, wonderful, smart, brainy us humans, are nothing more than modified great apes. Modified great apes. Our brilliant brain, our extraordinary brain that has given us control of the planet and produced all sorts of aberrant creatures as well. I'm talking about some of our politicians at the moment. Our brilliant brain is just a modified great ape brain. That's extraordinary that a single simple idea can explain so much, and so much that is important. So it's hardly surprising, you've probably seen this quotation before, famous evolutionary geneticist from the 20th century, Theodosius Dobzhansky, quipped, "Nothing in biology makes sense except in the light of evolution." And again, I've got a nudibranch, a weird, wacky sea slug to enshrine the products of this great process. What does that mean? It means you cannot approach a biological problem without taking into account the evolutionary process. Let me explain. Let's say you're interested in a membrane pore. You're doing detailed biophysics to understand the structure of the pore. You've measured, the 3D structure of the proteins in question. Everything's down to literally angstrom unit distances and so on. And you're measuring the rate of trafficking of ions across the pore and so on. You're doing exquisite engineering science in a sense. And you can think of this as a solution to an engineering problem. This is the solution to the problem of getting ions from point A across the membrane to point B. So yes, it's completely legitimate to think of it as a problem-solving exercise, to bring a engineering mindset to biology. But the difference is this, that membrane pore is not designed by engineers for a particular purpose. What it is, it's something that evolved, entered the tree of life, if you like, a long time ago, and it's probably as some other transmembrane process, and that over time mutations have occurred. It's been tweaked and 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 tweaked over the generations. So there are two dimensions to your analysis of the membrane pore. One is, yes, engineering, functional. But the other has to be historical, because history informs the way this thing works. And it's maybe best to think of this in terms of analogy. Let's say there's an old, rotten plank hut on the side of a small river, okay? And something changes. We now need to bridge the river. We don't need the hut anymore, okay? So we've got, in biological terms, this is a change of function, okay? What would I do if I'm an engineer? Well, I need to build a bridge. I'd bring in some I-beams, some girders. I'd pour some concrete. Right, easy, okay? Not if you're stuck, if you like, with history, because you've got the materials for your wooden hut. So you've got these old planks, these old pieces of wood, which you then reassemble into a bridge. It's not the best possible bridge. It's not the best possible engineering solution to the problem, but it's the engineering solution you have to use because it's predicated on what's already there. You are changing what is already there. You are building on history. Okay, so that's why this idea, as Dobzhansky says, is basically central. You cannot think biologically without incorporating that kind of historical perspective into your thinking. And the way we've typically thought historically is through inference. What do I mean by that? Well, what we do is we look at the distribution of traits across individuals, or in this case across species, and we can make inferences. As I say, we can travel back in time sort of conceptually by looking at the distribution of traits across, in this case, our family tree. So notice the green trait is present in species 1 and 2, but individuals 1 and 2 in species 1, but not in 3 and 4. In other words, it must have arisen within species 1 in the common ancestor of individuals 1 and 2, after the common ancestor of individuals 1 and 2 split off from the common ancestors of 3 and 4, okay? And vice versa, in species 2, all 3 individuals share the purple trait, the purple mutation or whatever it is. What does that mean? Here it is. It must have arisen in the common ancestor of all 3. See what we're doing? We're taking contemporary distributions to make inferences about the past. And for a long time, and that's a powerful process, and we're pretty good at it, and we've derived a lot of interesting information about it. What I want to talk about today is the fact that new technologies have changed everything. We no longer have to try to reconstruct the past by looking at the present and making inferential arguments like this one. Now we can travel into the past. And this is so cool. This is using ancient DNA, so-called ancient DNA, as a time machine. So I'm sure you're aware of the technologies surrounding ancient DNA. These have now been with us for about 15 years. There are multiple problems of working with ancient DNA. The big one, of course, is that DNA falls apart fairly slowly. It's a fairly stable molecule, but it does degrade over time. So you do not have nice long strands of intact DNA. You've got lots of little bits of DNA from your sample. Obviously, as I say, the quality is going to depend on all sorts of things, like how it was preserved, what the conditions were, and so on. There are all sorts of ancillary problems like contamination. All sorts of things can happen to your bones or whatever you're trying to recover your DNA from posthumously. And there's complications too about the material that you're using, because obviously you're somewhat destructively sampling. So if you've got a precious archaeological specimen, it's possible the archaeologists won't be too excited about you drilling little holes in it in order to recover material for your ancient DNA analysis. And there are limits to this again. This is dependent upon the rate of degradation. I put here on this slide that the sort of maximum is probably around 1 million years old. That's generally true. There has been a study which claims to have recovered usable DNA sequences from about 2 million years. That was in permafrost in the Arctic, though. So those are indeed extreme and very helpful preservation conditions. But yeah, we can't travel, tragically, we can't travel with our DNA time machine all the way back to the dinosaurs. We're looking at, within the past million years or so in practice. Now, given the fact we're especially interested in our species, Homo sapiens, that's fine, right? Homo sapiens, it's a little bit open to debate exactly when you say Homo sapiens appeared, but, certainly more recently than 1 million years ago. So arguably, ancient DNA gives us access to our entire human history. Now, the reason that we can do this is it's a byproduct of so-called next-gen sequencing technologies, which are super fast, super efficient, and super cheap. Okay, so in other words, before these technologies came along, the fact, yes, you've got some ancient DNA, but it's lots of tiny fragments, tons and tons and tons of tiny fragments, meant it was basically unworkable. Now you just sequence every single one of those tiny, tiny, tiny little fragments, and then you can crank them through some big bioinformatics analysis package, which will glue them all together looking for overlap. And so you're going to get hopefully as close as possible to a total genomic sequence, with the help, as I say, of an awful lot of sequencing and a lot of computation. Obviously, it helps if you're dealing with something like Homo sapiens, where we have reference genomes and so on. So it's not just a matter of looking for overlaps. We're building on a scaffold of a pre-existing sequence and a well-understood set of chromosomes and so on. But anyway, that is the technology. Technology is a technology based on these new sequencing and computational technologies, and it has changed the way we think about ourselves, and in some ways about how we think about the evolutionary process. So those are two pretty big claims. It's changed the way we think about ourselves and about how evolution actually happens. So let's investigate those claims. Here is the key point. We're back at exactly the same situation as before, but now we have ancient DNA, so we can travel back in time. So here's our time machine. So say we're interested in what happened up here. Woo! I need a sound effect for that, obviously, but you get the general idea. Now we can actually go and sample that population if we've got appropriate specimens and so on, and maybe even over time we can track the change in allele frequency for the particular mutation. This is a favored mutation undergoing positive natural selection. So that's what we're going to do. And the material we're going to use, this is the famous sort of canonical case that put ancient DNA on the map, is from Siberia. This is a Denisova cave in which there's a whole slew of material. As you see, it's in southern Russia, down here. But most critically, there was a tooth and a finger bone, which yielded reasonably high quality DNA, and from which we were able to infer, and I'm sure you're familiar with this, a whole new member of the human family tree. A whole new member of the human family tree, for which we have well, we have one little finger bone and a tooth. That's it. So it's not the normal situation where we're adding members to the human family tree, where we're finding fossils and we can reconstruct them and so on and so forth. This is a DNA-based new family member. Turns out to be closely related but distinct from our established closest relative, which is Homo neanderthalensis. Homo neanderthalensis evolved in Africa, and headed out of Africa on the order of 600,000 years ago, moved into Europe and Western Asia. And it seems that a population or a set of populations derived from that sometime early in the history of Neanderthals, the Denisovans, so called after their cave, went further east typically than Neanderthals, though they did cohabit certain regions and they did interbreed. Okay, so this is what I mean about changing the face of our understanding of ourselves. So we now, and this has been now known for some time, we now appreciate that we've had multiple instances of hybridization between modern humans and these so-called archaic species, so Neanderthals and Denisovans. This was actually from a fairly early paper reconstructing that process. It's still more or less accurate. The key thing here is if you are not a pure African, you almost certainly have a not insignificant amount of Neanderthal DNA in your genome. On the order of between 1 and 2% of your genome is derived from those hybridization events with the Neanderthals. The reason they're only in non-Africans, remember the Neanderthals and Denisovans were out of Africa. They were in Eurasia. So it was when our ancestors, modern human ancestors, came out of Africa. No, so these are the Africans that left Africa, if you like. They encountered these populations, they interbred with these populations, and the result, as I say, is that we are part Neanderthal and indeed varying from population to population, part Denisovan. Okay, I think this is a stunning recalibration of our history. We've tended to think of human evolution in the past as a sort of grand march from that great ape ancestor to us, wonderful, brilliant us, right? It's a march involves bipedalism, involves evolving a huge, big, super brain. It involves all this wonderful manual dexterity, tool use, involves changes in the structure of the throat, so we've got language. But you get the idea, it's this sort of vectored change. Everything is moving towards the great sophisticated uber being that is Homo sapiens. Well, I mean, yes, to some extent those things happened. Yes, I don't think they happened in any particular vectored way. I mean, we now know, for example, that bipedalism, we know this from the fossil record specimens like Lucy, bipedalism evolved fairly early and then brain evolution followed. So Lucy, who was about halfway from the point at which our lineage split off from the chimpanzee's lineage about 7 million years ago, Lucy, Australopithecus afarensis, dates to around 3.3 million years ago. So literally about halfway between our common ancestor with chimpanzees and us. She was fully bipedal, but had a chimpanzee-sized brain. So the brain evolution piece was in the second half, you might say, of hominin evolution. So the vision is no longer that vectored march. It's this messy process involving furtive sex behind the bushes in the Middle East when our ancestors first met the Neanderthals, more sort of weird interactions with the Denisovans and so on. It's not this clean, clear march. It's a story of weird sexual hybridization in unusual places and so on. Okay, it changes everything. But I want to focus on two particular features of the ancient DNA story pertaining to humans, which we can now infer. One is, when did this hybridization event occur? I mean, I've loosely told you it was as our modern humans came out of Africa, but can we do better than that? The second one is, what did we get from Neanderthals? What's in that 2%? So let's start with the when question, and here we're going to go back to Genetics 101. And just to remind you about what happens in meiosis, you get exchange between non-sister chromatids of homologous chromosomes, mum and dad chromosomes. So here we have the chromosomes lined up. Here's the crossing over event, and here's the outcome. So these are going to turn into 4 gametes. So you've got, these are the outcomes of the recombination, recombinant chromatids. Okay, now, fine, simple basic process, which I was going to say came to us from Mendel. It didn't come to us from Mendel, it came to us from Sturtevant and Morgan with the early years of fruit fly genetics. Now, how is this relevant to our problem, and how are we going to use our time machine, ancient DNA, to address this issue? When did the hybridization occur? Well, the answer is, well, let's just track sort of theoretically the introgressed, i.e. introduced Neanderthal DNA in a modern human genome historically. So here's the first, we're going to focus on one chromosome. Red is Neanderthal, blue is modern. And what we're going to do is have a hybridization event. So obviously we've got our 2 chromosomes, the maternal and paternal chromosomes. These red are Neanderthal, say blue is modern. That, by the way, is our symbol of modernity, is the statue of John Harvard. Okay, so now we've had our hybridization event. We've got a full heterozygote. One chromosome is Neanderthal derived, the other is modern derived. Now, let's just, as I say, this theoretical scenario, let's assume the moderns are surging forward, so everyone else in this immediate vicinity, the poor old Neanderthals, have been sent back into the wilds somewhere. So what we're going to do is now effectively back cross this hybrid to another modern. Okay? Now, what's going to happen in the generation of the gametes in this heterozygotic individual? What is going to happen is, so here's, again, we're just focusing on this chromosome, we're going to have crossing over, right? So what does that result in? So it means that the gametes are going to be chimeric. They're going to be partly, for this individual, they're going to be partly modern and partly Neanderthal, okay? So the next generation, that's what we've got. Now we've got one hybrid chimeric chromosome, and the other is a pure modern chromosome. Let's do another cycle of that. So this is another back cross into the modern population. What's going to happen? Well, you're going to have more recombination. Now, of course, recombination is occurring, by the way, on the blue-blue situation, but it doesn't make any difference because it's all blue. So you're just swapping bits of blue to other bits of blue. But here we've got more separation of the red and blue segments in the hybrid situation. So now let's look at that over the generations. So we've now got 3 generations of individuals here. One starting with the first hybrid offspring, then the grandchild, and then the great-grandchild. And the take-home here is obvious, that the segments of red are getting smaller each generation. They're being broken up by recombination. Okay, so good, we've got that. How does that help us in terms of looking for the timing of the events associated with the Neanderthal modern hybridization event? Let's look at some data. So the blue blob on this map kind of shows you the general distribution of Neanderthals. And this is a study, an ancient DNA study, which is acquiring material from these various locations. Okay, fine. Now, let's look at material from this cave, the Bachakiro cave, from about 44,000 years before present. So this is an individual whose genome has been sequenced, and we can then go into that genome and ask a simple question: where are the Neanderthal-derived bits? Because we know what the Neanderthal sequence is. And what we find, obviously there's plenty of small segments, but also find a whole bunch of big slabs, relatively big slabs of Neanderthal DNA, okay? So that is telling us, to think back to the model I've just presented, that this must have come relatively recently after the hybridization event, because we haven't had that much, well, we've had a fair bit of breaking up of the Neanderthal DNA, but we've still got some pretty big chunks. Now, this is the time machine. It turns out there's other materials, skeletal material from the same cave, which is more recent. This is 35,000 years ago. So what is it? That's 9,000 years later. Now look at the largest sizes of the blue on this chromosome. So we've broken this down just by chromosomes. And yeah, there's plenty of blue, still plenty of blue, less blue because we've lost some of the Neanderthal segments, presumably partly down to drift, partly down to maybe selection acting against them. But you've still got plenty of blue, but none of it in big slabs. Everything's broken up. So this is exactly according to the model I've given you. Over time, these segments are going to break up, and we can take it further. We can actually then, I mean, it's model based and assumption based, but we can come up with an estimate that the hybridization event occurred at a certain time prior to the individuals that we're sequencing. I think that's super cool, that we can track the diminution, the decrease in size of Neanderthal-derived chunks of DNA in ancient genomes, literally traveling in time and watching them come down in size, and from that extrapolate backwards, if you like, to the timing of the original hybridization. Second thing I want to talk about is, what did we get from humans? So it turns out that the DNA that we have, if you look across all of us, there's a fair bit of the Neanderthal genome which is missing, so it's just not present in modern humans. So presumably that was deleterious. It was eliminated by natural selection. And then, well, so here's my genome, and here's the overlap with the Neanderthal genome, and here's your genome, and here's some overlap. So there's quite a lot of different pieces of Neanderthal genomes floating around in different people's genomes. What I want to focus on are these ones, the ones which, if you like, overrepresented in modern human genomes. So they're much more frequent. This segment is much more frequent than you would expect by chance alone, and certainly more frequent than other segments. So what it seems to be is there's been what I call genetic cherry picking, that certain features of the Neanderthal genome are actually beneficial. And if you think about it, this makes lots of sense, and again tells us about how human evolution occurs. So Neanderthals came out of Africa, or Neanderthal ancestors came out of Africa. They were African. They're moving into Poland, let's say. Completely different climate, much colder, completely different pathogens. You're dealing not with tropical diseases, but with diseases in the temperate zone and so on. So they have to evolve. Now, let's take a simple example. They're in a much colder place, and they're getting much less sunshine. So they probably want to be less dark-skinned, because being too dark-skinned in a place without very much sunshine means you don't generate enough vitamin D. Okay? Now, so what's happened? Yes, we know that Neanderthals were quite fair-skinned. So how did that happen? Well, it meant some dark-skinned individual had a mutation in a gene involved in pigmentation, which resulted in an offspring who was slightly lighter, and that one did slightly better because it got more vitamin D and therefore had more children. This is natural selection, right? Had more children which carried the same trait, and those were passed on to the next generation. And each generation you're getting a slightly higher frequency of the fair skin allele. Okay, that's natural selection at work. It's also incredibly slow and clunky and inefficient. So what happened with modern humans? They had to adapt in the same way. They too are Africans coming out into this alien, hostile environment. But instead of having to go these generations and generations and generations of natural selection, what they did was they had sex with Neanderthals, and they got a cassette of pre-made adaptations. The adaptations, like immune and pigmentation traits, which are the adaptations that the Neanderthals had spent 500,000 years evolving in response to the European environment, moderns, courtesy that introgression, that hybridization, acquired those. Super cool. Beautiful example of this. If a weird one. Tibetans, as you may know, are different from the rest of us in that they have evolved to thrive at the super high altitude, 4,000 meters of the Tibetan plateau. Whereas you or I, assuming you're not Tibetan, if you take us up there, we're gasping, we can barely move, we're pathetic, right? It turns out we know the gene that is involved, or a major gene that's involved. This is on the basis of genome-wide association studies and so on. It's a gene called EPS1, and it's involved in oxygen metabolism, surprise. And that seems to be the key to Tibetan success in this regard. Fine, cool. The weird thing about EPS1, it's not just a little bit different from my EPS1. It's not a little bit different from other Asians' EPS1. It's many, many mutations different. It's like a quantum leap different from other humans' EPS1. How did that happen? Let's dig into the details. This is a slightly complicated plot. I apologize, but it's actually super illuminating. So on the x-axis, for want of a better term, we've got, this is just the DNA sequence of the EPS1 gene. And then on the y-axis, vertically, each line is an individual. So we've got about 50 Han Chinese, so in other words, non-Tibetans from the same vicinity. Then we've got another 50-odd Tibetans. Now, it's a bit confusing. The background gray is default sequence, and black shows differences or distinct particular mutations, okay? And what I want you to see is three things. One, there's pretty much a sort of, it's a bit messy down here, it's a bit messy up here, but basically we're looking at a sort of standard Han Chinese sequence across here, and the same here. The Tibetans have a standard sequence. Now, the key thing is that this Tibetan sequence note is very distinct from the Han Chinese one. So this is what I mean about the quantum leap in terms of difference between the two. The killer is this. I think this is super cool. Here in green up here at the top left, we have a Denisovan sequence. So it's not identical, but what we see is essentially the Tibetan sequence is the Denisovan sequence. So what does that tell us? It suggests that Tibetans, or at least the ancestors of Tibetans, acquired that EPS1 allele with its adaptation to high altitude from the Denisovans. So this is the adaptive introgression of a beneficial allele from one of those early hybridization events. So what does that look like? So there's a slightly complicated plot, but this is the sort of recent part of the human family tree. So here's starting 600,000 years ago, we're still in Africa. The split occurs between Neanderthals, Denisovans, and moderns, okay? And the Denisovans and Neanderthals are on this lineage, and here's a split between Neanderthals and Denisovans. Now, what I've done is I've color-coded this sort of according to genetic similarity. As lineages progress through time, they're going to acquire separate mutations and become genetically distinct. So obviously, very recently after the split here, we're still pretty purple, but as time goes on, the Neanderthal lineage is becoming genetically distinct, and so too is the Denisovan one, and so too is the modern human one, okay? Now, at some stage, you have a split off with the population, it's very recent, into Tibetans, a population that is moving into the Tibetan plateau, presumably. And obviously, so this is all in keeping with the green line of modern humans until you get this hybridization event. So this is where the Denisovans, or ancestral Tibetans, the Denisovans have seeded, so to speak, through hybridization, the ancestral Tibetans with the Denisovan allele. So this, for this gene, and this gene alone at least, Tibetans have the red sequence, i.e. the Denisovan one. Amazing. As I say, this changes. It's a game changer for how we think about how the evolutionary process works. This is not one mutation at a time, natural selection, one mutation at a time, natural selection. This is a whole slab of mutations acquired through, as I say, hybridization with another population, another species, which has adapted to that environment over the past 500,000 years or however long. Fine, super cool, really interesting. Problem. We only had material for Denisovans from the Denisova cave, right? The Denisova cave is at about 700 meters above sea level. It is not at altitude. So why on earth would Denisovans go to all this trouble to evolve a high altitude allele if they lived at low altitude? That was crazy. It doesn't work, and that's not how natural selection works. Obviously, it's not like natural selection would prepare the low-living Denisovans for a future move to high altitude. So there's a sort of weird glitch in the story. But that was resolved fairly recently when we found the second tranche of Denisovan material. It's half a lower jawbone. Here it is. Unfortunately, not good enough preservation to yield any DNA, but it has morphological distinctness, which strongly suggests Denisovan. This is the cool part. Where was it found? It was found here in a cave in Tibet at about 3,500 meters above sea level. In other words, the Denisovans most certainly did inhabit ancestrally the same regions as modern Tibetans. They evolved to withstand the pressures of living at those low oxygen partial pressures, and they were also on hand, presumably, to hybridize with the ancestors of modern Tibetans as they moved into Denisovan territory. So there it is, a modern story of adaptation, which isn't done through a stepwise, what it was for the Denisovans, a stepwise process of natural selection. But here, the ancestral population, in this case the Tibetans, have acquired wholesale the adaptation through a hybridization event. Let's say, ancient DNA is telling us all sorts of really cool things. It's rewritten the prehistory of humanity. It's rewriting the history of humanity as we learn more about the movements, invasions, and diaspora of modern human populations. But it's also giving us a new vision about how the evolutionary process occurred. Evolution is central to biology, and it's forever yielding new and important insights. Thank you for listening.